Hello everyone, this is a brief explanation on applied immunology. I'll be presenting on two main points, which are I'll be presenting first on the cooperation between T and B cell activation, and also that one has three questions that would need to be presented. Then also on principles of vaccination also has three questions to be presented on. So before going further, I would like to first define what applied immunology is. Applied immunology is a subtype of immunology that focuses on the practical application of immunology in medicine. It supports the development of biological therapies such as vaccine, diagnostics, and prognosis. So, I also define what cell activation is. Cell activation is the process initiated by binding of a ligand to its receptor, which enhances the cell's ability to respond to various stimuli. This is where we have two types of activation, which is the T cell activation and the B cell activation. So, question 13 that says, how do T helper cells support B cell activation? So, T helper cells, it plays a crucial role in the activation of B cells. And these B cells are responsible for what? For producing the antibodies. So, this interaction is essential for adaptive immune responses, that is particularly in humoral immunity. So, the support provided by T helper cells involves several ways. These are the steps how the T helper cell supports the B cell activation. One, we have antigen presentation by B cells, we have activation of T helper cells, we have cytokine secretion, we have B cell differentiation, and also the CD40 and CD40L interaction. So this is the brief structure to give us a highlight explanation about how the helper T cell activation and its action in the B cell activation. So one, where we said in the antigen recognition, the antigen it can recognize which are the effector cells, it can recognize the MHC2 and the CD4 plus only. And in then it will go to the clonal secretion, which is here that the T helper cells help us also in what? In the memory T cell. So in the third one, that is the interleukin secretion, here the effector cells come into the neutrophils and macrophages, which are the non-specific defense in the B cell activation. We also have the killer T cell, which is in the cellular immunity. And also we have the B cell, which helps in the, now it will come to the humoral immunity and give support to the B cells. So question 14, which says, what is isotope switching and how is it regulated in the immune system? So isotope switching, first, its definition, it's when a B cell is activated and it produces hemoglobin M antibodies, which are the first line antibodies in response to an infection, where we all know that hemoglobin M, it helps us it's the first line of antibodies and it helps us in response to any infection that comes across us. So this type of switching is a highly regulated process that involves several key factors. The factors that can involve how isotype is regulated. So one, we have the B cell activation by antigen and helper T cell signals. We have second, the CD40 and the CD40L interaction between B cells and T helper cells. And also we have the cytokine signaling from T helper cells, which is we have the interleukin 4, 5, TGF beta cells, and etc. that drives the switching to the specific isotypes. We have the activation induced cytidine deaminase, introduces the DNA breaks in the switch regions of the immunoglobulin high, heavy chain locus. The heavy chain locus recombines, resulting in the expression of a new immunoglobulin isotype, which is IgG, IgA, IgE, and etc. In question 15, we have what are T follicular helper cells. So I'll be defining what T follicular helper cells are. So T follicular helper cells are specialized subtypes of CD4 T helper cells that plays a crucial role in supporting the B cells responses. They actually just plays a role in supporting the B cell responses, since we are talking about the interaction between the T helper cells and the T cell activation and the B cell activation. So question 16, we have what makes a vaccine effective? What is the effectiveness of a vaccine, right? So an effective vaccine is one that stimulates a robust, durable, and protective immune response that prevents infection and reduces the severity of diseases. So here are the key aspects that determine how well a vaccine works. For you to know that this vaccine works well, these are the aspects you need to follow. The first one, we have the antigen, where the vaccine must present the high antigen to trigger the appropriate immune responses. Second, we have the immune action or the immune activation. It should stimulate both humoral antibody and mediated cell immunity. We have the memory, where the vaccine must provide long-term immunity by creating memory cells. We have the adjuvants and formulation. Later on, I will be defining what adjuvants are to enhance the immune responses and ensure stability. We have the safety, where the minimal side effects to ensure widespread acceptance for this patient. We have many other aspects like efficacy and effectiveness. We have herd immunity, we have global accesses, and so on. So the question 17, the one before the last, that says, how do messenger RNA vaccines work? How do they work? So messenger RNA, this vaccine represents a novel approach to vaccination that uses messenger RNA to instruct cells in the body to produce a protein that triggers an immune response. So here are the steps by step of how mRNA vaccines work. One, mRNA is injected into the body. This is the first step. Second, cells use the mRNA to produce a viral protein, spike protein in COVID-19. Step 3, the immune system detects the foreign protein and activates an immune response. 4, the immune system creates memory cells to recognize and respond quickly if exposed to the pathogen in the future. 5, the body clears the mRNA and protein. So we have also some importance of vaccine, that mRNA vaccine. The importance of it is speed of development, and it also has no risks of infection. You cannot be infected, and it also there's no need for adjuvants, and also flexibility for new pathogens. So this is a highlight structure of how messenger mRNA vaccines work, where we have first the mRNA packages into the lipid nanoparticle. First, the second mRNA enters the cell. Instruction for making spike proteins are passed to the cells. 3, the cell makes protein that leak out of the cell and create spikes. 4, white blood cells recognize spike proteins and makes antibodies. 5, antibodies attach to virus and block them from affecting the cell. It cannot infect the cells. This is how messenger mRNA vaccines work. So the last question where we have, why are adjuvants used in vaccine? First, I would like to define what adjuvants are. Adjuvants are substances that are added to vaccines to enhance or boost the body's immune response to the vaccine's active ingredient, usually the antigen, and the substances that stimulates immunity. This is adjuvants. So here's the breakdown of why adjuvants are used in vaccines and how the importance of it. So 1, it helps us in enhancing the immune responses, where stronger and more durable immunity. Adjuvants stimulate the immune response in a way that enhances the body's immune to the antigen. 2, second, we have it reduces the amount of antigen needed, whereby there will be lower antigen dosage. We have broadening the immune responses, where we have the increased immunity across different age groups. We have prolonging the immune responses. We also have enhancing the activation of memory cells, where we have it helps in boosting the memory cell activation or formulation. We have in providing vaccine efficacy in different populations. increase efficacy in the elderly. The immune system of elderly individuals tend to weaken with age. So this efficacy in different population can help this elderly age to increase their efficacy. Adjuvants in combination with mRNA and subunit vaccines, and also it helps in the safety and consideration. While adjuvants are generally safe and play an important role in enhancing the immune response, they must be carefully selected and tested to avoid unwanted side effects.